Hello, everyone. My name is Alejandro, and my topic is therapeutic approach for aberrant RNA splicing. Some background info would be that eukaryotic transcripts are processed to create the mature form of mRNA for proper translation through different mechanisms. The first one being the 5' cap, which is a 7-MEG linked to the 5' end through a 5' triphosphate linkage. The next mechanism is the 3' poly A tau, which is created through cleavage of the poly A addition site. And then the enzymes PAP and PABP give rise to the tau. And these two mechanisms have functions in relation to preventing degradation, promoting translation, and RNA transport. And they also function in splicing mechanisms. And the final mechanism is RNA splicing itself. Why did I choose this specific topic? Well, there's various diseases that arise due to splicing mistakes. And I was curious if there might be some sort of mechanisms that fix these mistakes. And it just so happens that there are some gene therapy techniques that have been developed to fix these mistakes. Some additional background is that by the manner of alternative splicing, different mRNAs and ultimately different polypeptides can arise from a wide variety of various combinations of exons by primary transcripts being spliced together in a single gene. And most pre-mRNA splicing is done by the spliceosome, which has 5 snRNAs, which are U1, 2, 4, 5, and 6. And its main manner of functioning is shown in this figure to the right, in which the 2' OH branch point attacks the 3' OH of the 5' splice site. Then the 3' OH of the released exon attacks the phosphate at the 3' splice site. And this ultimately joins the two exons together. Some more background on the splicing mechanisms that we learn in this class would be the self-splicing introns, which the first one is group 1 and is shown in this figure to the far left. And it basically utilizes the 3' OH of a GTP to attack the 5' end splice site. Then the 3' end of the 5' end splice site attacks the 3' splice site and ultimately completes the reaction. The next group is group 2, and it's shown in the center figure. And it's basically works in a similar fashion to the spliceosome, but it depends more on the RNA structure to create the active sites. Then the last mechanism that we learned is the trans-splicing, and it's shown to the far right. And it basically functions by having exons from different RNA molecules fused together. And it works by having the branch point on the pre-mRNA attack the 5' splice site of the slRNA. And then the 5' splice site then attacks the 3' splice site, forming the newly formed mRNA molecule. Now, the diseases that arise from mistakes in this splicing process include changes in the splicing signals, which are the 5' and 3' splice sites, or mutations in the splicing machinery itself. And the therapeutic approach to these mistakes is through gene therapy, the first one being antisense oligonucleotides, which are short oligonucleotides guided to the pre-mRNA, and it targets splice sites to modify the splicing reaction and ultimately leads to mRNA repair and protein function. And its main form of mechanism is shown in this figure in part A, where it's basically showing that it's sterically blocking motifs in the pre-mRNA without promoting degradation, while at the same time shifting the splicing outcome. Now, the next gene therapy technique is the spliceosome mediated RNA trans splicing, also known as SMART. And it basically utilizes the spliceosome, target mRNA, and pre-trans splicing molecules, which are known as PTM. And it's shown in part B. And it basically is showing how it functions by introducing a section of repaired mutant transcripts with the main goal of recombining endogenous mutated target pre-mRNA with exogenous PTM to achieve substitution of mutated regions for the wild-type sequence, ultimately fixing the mistakes. The last gene therapy technique is siRNA, and it functions by silencing the expression of genes by preventing translation through promoting degradation due to the base pairing that it does. And this is shown in part C, and it's basically showing that the siRNAs are targeting aberrant splicing isoforms, and it's promoting the degradation of these incorrect splicing that occurred. Some lingering questions about the topic would be that the manner of delivery includes viral and non-viral methods. The viral methods have a higher transfection efficiency, are marked for immunogenicity, phenotoxicity of the transgene also occurs, and there's a possibility for vertical and horizontal transmission by replication-competent viruses, whereas for non-viral methods, it has a low immunogenicity, but as an effect has a low transfection efficiency. My question is, which method would be best suitable for utilization across all patients, not just a select few? So basically, which one works best for any type of patient? And these are my citations, and thank you for listening to my presentation.